Student in Dr. Lüberstedt's lab. And today I'm happy to introduce our final speaker, Noé Perron. Noé Perron is a second-year PhD student in Dr. Matthias Kürst's lab in the Plant Molecular and Cellular Biology program at the University of Florida. Before joining the University of Florida, Noé obtained his master's degree in Agricultural and Environmental Sciences from the Agriculture Engineering School Isara Lyon in France. During his time at Isara, Noé participated in the Erasmus Plus program, where he spent time at the University of Budapest in Hungary, studying plant physiology, genetics, and wildlife conservation. Noé is a very active graduate student who participated in multiple graduate organizations at the University of Florida. He has been awarded with a Fulbright scholarship from the University of Florida Carl Steens Award. Currently, Noé's research is focusing on investigating the genetic basis of drought and salt stress adaptation in common ice plant, investigating their photosynthetic adaptation to abiotic stress, and the transition of photosynthetic mechanisms from C3 to CAM plants. The title of his presentation today will be Single-Cell Study of C3 to CAM Transition in the Ice Plant, Points to New Strategies for Increasing Drought Tolerance. Please join me in welcoming Noé Perron.
Thank you very much for the nice introduction. Good afternoon, everyone. I'd like to start by thanking the organizers of this symposium, not only for inviting me to speak today, but also for providing this great platform to meet people and exchange great ideas. I think the day has been excellent so far, so thank you for that. I'm very excited to be here today to talk about our single-cell study of the C3 to CAM transition in the ice plants and explain how it points to new strategies to increase drought tolerance in crops. It's a fascinating topic, and to understand better what all that means, I will start by giving a bit of background information. The increasing occurrence of drought events threatens the sustainability of our agriculture. While a short-term solution to that issue could be to temporarily increase crop irrigation, this practice will only further aggravate the current freshwater crisis. In this presentation, I want to show that we can fix this and without using more water. To find an appropriate solution, we need to look at what is the main driver of productivity in the farm. I am talking about photosynthesis. The vast majority of plants on Earth perform C3 photosynthesis. It is a pathway that is characterized by the opening of stomata during the day when the temperatures are high and the evapotranspiration are high too. So most of the water during that time goes out of the cells. But during the day, CO2 flows inside of the mesophyll cells, which is where the bulk of photosynthesis occurs, and is carboxylated by rubisco, which fixates CO2 inside of the reductive pentose phosphate cycle, and this ultimately leads to the production of photosynthetic sugars. During the night, the stomata of C3 plants close. So the issue with this pathway is that it does not allow plants to conserve water, and so it does not allow plants to survive long periods of drought. But some plants, like cacti and pineapple, evolved a new mode of photosynthesis called the Crassulacean acid metabolism, or CAM, which allows them to conserve water and survive long periods of drought. CAM plants achieve this by opening their stomata during the night when the transpiration rates are very low, so most of the water can stay inside of the cells. So CO2 is assimilated during the night and carboxylated this time by phosphoenol pyruvate carboxylase, or PEPC. And this results into the accumulation of C4 acids inside vacuoles during the night. CAM plants have very large vacuoles. During the day, the stomata of CAM plants close, so the transpiration rates are high and the water stays inside of the cell. However, CAM plants are very hardworking and work both the night and the day shift. During the day, the C4 acids are transported to the cytoplasm to be decarboxylated, and this provides the substrate CO2 for fixation inside of the reductive pentose phosphate cycle. So this part is common with the C3 plants, and the cycle continues. We know that CAM plants achieve 3 to 6 times the water use efficiency of C4 and C3 plants. So a great strategy to increase the drought tolerance of crops could be to introduce some elements of the CAM pathway into these crops. Now that we've been through the details of CAM, it is time for me to introduce the fascinating system that is Mesembryanthemum crystallinum, or the common ice plant. And it is so fascinating because it is a facultative CAM species, meaning that it is a C3 plant most of its life cycle, but it has the ability to switch temporarily to CAM when it is subjected to drought or salt stress. We know that this transition occurs after 8 days of salt treatment. Salt treatment is used to induce physiological drought, but we don't know much about the main regulators of this transition at the molecular level. This is why the question that drives my research is the following: can we identify the regulators of CAM induction by single-cell genomics analysis? The best way to answer that question is by using single nuclei RNA sequencing technologies. So I will now try to explain briefly how this technology works and why it is so great for this kind of project. Let's start with a very simple question. How much do American people like IPAs, which are those very bitter beer, often a bit stronger than average, but also very tasteful ones? There are two approaches, two ways to answer that question. In the first approach, we're going to ask many American people from every state to rate how much they like IPAs on a scale from 0 to 5, 5 being I really love IPAs. Then we're going to calculate the average of all the answers, and we're going to apply it to the entire country. So this will be the value that defines the taste for IPAs of American people. As you can see on this example, we can draw the conclusion that American people really like IPAs. The average is somewhere between 4 and 5, probably. However, this is only informative for studies that assume that all Americans are a homogeneous group. But this is useful, for example, if you want to compare the taste for IPAs of American with the taste for IPAs of French people, for example. And this is the kind of depth you get with bulk RNA sequencing or traditional RNA sequencing. There are a few problems with this approach. The first one being that the representation can easily be very far from reality. And it's hard to know what people in smaller states think of IPA because the average will be mostly driven by the highly populated areas. And of course, smaller states matter too. This is why there is another approach. In this second approach, we're going to take the same results, but this time we're going to calculate the average per state. So we're going to consider that each state is one entity. But these are the same results, and we can already see that the reality is actually much more complicated than how it was shown by the previous approach. People from very different states can have drastically different relationships to IPAs, and we can see actually that the high average was driven by the highly populated states like California, Texas, Florida. And this is the kind of depth you get with single-cell RNA sequencing, where the different states would be cell types which are parts of the same larger tissue, but where each cell type can respond differently to a same stimulus. And it's interesting to see that people in Iowa seem to actually really like IPA, so we'll see if that's true tonight. How can this technology be used for plant breeding? Like traditional RNA sequencing techniques, it can be used to identify markers for desirable traits that drive some phenotypes of interest, but this time at the single cell type level. And this is important because if we're going to introduce markers inside of crops, we will want to be able to minimize the pleiotropic effects because we know that a gene expressed in one cell type can have a very different function when expressed in a different cell type. And of course, single cell also allows us to have a lot more power to identify these markers. This is a quick overview of the current lab projects that we have going on in the lab. So we are 3 PhD students, and we are all using single cell to answer different biological questions. So Jade Boyd for example, is using single cell to understand what drives nodule development in Medicago truncatula. Hank Schmidt is using it to understand what controls wood formation in poplar, and I work on the regulators of CAM photosynthesis. So let's get back to our project. Our hypothesis is that we can use the power that single cell or single nuclei RNA sequencing has to capture cells at different moments in their development, and by applying it to the C3 to CAM transition in the ice plants, we could be able to end up with a cluster of cells corresponding to the C3 performing mesophyll cells and a cluster with the same cells but in their CAM states. And by following the trajectory that goes from one cluster to the other one and looking at the gene expression in these cells in the middle, we could be able to identify some of the regulators of this process of CAM induction. Okay, now that the objectives are clear, let's get into the experiments. We watered 4-week-old ice plants with 0.5 molars of a common nutrient solution, and the same number of ice plants with the same solution, with the addition of sodium chloride to induce a physiological drought and to trigger CAM induction. After 8 days, these plants perform C3 photosynthesis, these ones CAM photosynthesis. We collected leaves from plants in both groups, both after 1 hour into the day phase and after 1 hour into the dark phase, because the CAM cycle occurs during both phases. We then isolated nuclei from each of the 4 samples and loaded them on a 10x Chromium platform, which is what is used for single nuclei RNA sequencing. After some time, the data was processed with Cell Ranger and analyzed with software packages that are called Surat and Monocle 3, and the outputs of the data analysis are these UMaps. UMaps are these beautiful graphs where each dot that you can see corresponds to one cell, and each cluster is supposed to correspond to one cell type. So these graphs are great, but they only become informative when we actually become able to assign each cluster to one cell type. So we do that by looking at known cell type specific markers from the literature and looking at how they are expressed in our data sets. By using this strategy, we managed to annotate our UMAP with the key leaf cell types. So we found that the mesophyll cells, for example, are shared on both sides of the UMAP. The 2 clusters at the bottom corresponds to the epidermal cells. We found 4 clusters corresponding to the vasculature. So for example, we have the xylem cells in 12 and the phloem companion cells in 9. Cluster 17 corresponds to the guard cells, very few cells in that cluster because guard cells are a very rare cell type, and replicating cells in cluster 14. So here on this slide, we have the combination of all 4 samples. So we almost have 23,000 nuclei, but it's also possible to look at the UMAP for each sample separately. And it's actually very interesting to do that because there are some very key differences in cell type composition, and it's interesting to see. So let's take a closer look at these differences. Let's start by looking at the differences between the day and night samples. As you can see in this light control sample, there's a cluster 4, that seems to have a lot of cells to it. But if we look at the same cluster, in dark control, we can see that there are very few cells there. However, if we look at cluster 7 in dark control, we see that there are plenty of cells. Same cluster in light control, close to no cells. And we have evidence that both cluster 4 and 7 are mesophyll cells that do photosynthesis. So this helped us to make the hypothesis and conclude that cluster 4 corresponds to the day mesophyll cells or photosynthesizing mesophyll cells, whereas cluster 7 corresponds to the night mesophyll cells or respirating mesophyll cells. Even more interesting are the differences between the salt-treated and the control samples. There is a group of cluster at the right of the UMAP that we know are mesophyll cells, and we can see in light salt that this group of clusters has many cells to it, but the same group in the control has very few cells. We can make the same observation when we compare dark salt and dark control. There are a lot of cells in this group of clusters, especially in cluster 3. If you look at cluster 3 in the control, there are almost no cells there. This is interesting. What will be good to do at this point is to look at how the main enzymes of the CAM pathway are expressed in this data set. So let's start by looking at the expression of phosphoenol pyruvate carboxylase 1, which is really the main enzyme of CAM. On this heat map, you can see the expression of the gene in the different clusters. So a high expression is represented by very bright colors. And you can see that the expression of this gene is, first of all, much higher in dark salt, which makes sense, but it seems to be localized to cluster 3 up there. And if we look at the expression in the same cluster, in dark control, we can see that the gene is almost not expressed at all. If we look at the expression of another CAM enzyme, an ADP-dependent malic enzyme, we make the same observation, a very high expression in cluster 3 in dark salt, a very low in dark control. And this is very interesting because we know that CAM photosynthesis is very active in dark salt at this point. So we made the hypothesis at this point that on the left of this UMAP, we had a cluster of mesophyll cells corresponding to the C3 performing ones, and a cluster of mesophyll cells, especially one cluster, that could correspond to the CAM performing mesophyll cells. To verify this hypothesis, we reclustered the mesophyll cells, so isolated them, and we basically reran the data processing steps. And we did that because our next objective was to perform trajectory inference analyses, which are used to uncover developmental trajectories based on single cell data. So at this point, we're basically doing what I showed during the objectives. We're trying to find this trajectory from C3 to CAM. So on this graph, you can see the output of trajectory inference methods. Each color corresponds to a different cluster. And so you can see the progression going from one cluster or one cell type to the other one. Traditionally, people will look at developmental trajectories from meristematic cells to a developed cell type, but we are going to see if there's something between the C3 and the CAM cell types. Because CAM is a single cell process, and we know that the CAM is biochemistry of the C3 cells is remodeled to accommodate CAM, as opposed as the creation of a new cell type to perform CAM. So let's see if it works. We found that there is a trajectory going from cluster 4, which is a C3 cluster, to cluster 2, cluster 2 to cluster 1, cluster 1 to cluster 6, and cluster 6 to cluster 3, which is our candidate CAM cluster. So this is very important because we found that there is an actual developmental trajectory going from our candidate C3 to our candidate CAM clusters. And this means that by looking at the genes that define the trajectory, that go on and off at different moments during the trajectory, we could be able to identify the main regulators of CAM induction. So this is where we are at right now. We are currently looking at these genes, so I'm going to be leaving you on a cliffhanger for now, but hopefully we'll have some good results to communicate in the next few months. But in conclusion, we showed now that the ice plant single-cell transcriptome provides the keys to achieving a broad understanding of the CAM induction process. Next important step will be to validate the cluster notation via Gus staining. And hopefully, further trajectory analysis may help identify the key regulators of CAM induction. I'd like to end this presentation by thanking everyone in the Forest Genomics Lab at UF for their constant help and support throughout this project. And of course, I'd like to thank Iowa State University for having me here today and the organizers of the symposium. Thank you very much.
Thank you very much, Noé, for your wonderful talk. And does anybody have any questions?
Yeah, I have a question. Why did you decide to look at nuclei instead of the entire cell? I'm curious if some of the organelle genomes would have an effect on it as well.
Yeah, this is a very interesting question. We tried, so it's very complicated to do single cell in plants because of the cell wall. We cannot sequence the RNA because the way single cell works is that it sequences the mRNAs directly within the cell or within the nuclei. And it's very hard for the enzymes to penetrate the the plant cells because of the cell wall. So what we have to do is to digest the cell wall to create a protoplast, basically. And unfortunately, this process, we know, induces a lot of gene expression that would completely bias our analysis. So we try to do protoplasting, and it also doesn't, we know that protoplasting also doesn't help to identify some of the rare cell types like guard cells, which were very important for us. So because of a lot of papers came out showing that single nuclei was actually more effective, more efficient, we decided to go with the nuclei. It is true, we don't worry too much about organelles. I guess the drawback of that method is that you cannot capture the mRNAs that are floating in the cytoplasm. That's very few of them. So hopefully we don't miss anything too important, but it is still something to keep in mind.
Interesting talk. So you're focusing now mostly on the induction of CAM. What would it take to really go to the whole mechanism? If you're thinking like, okay, can we transfer this to another C3 plant like rice?
Right.
Then you also have to introduce the whole CAM mechanism. Is that? Do you have any thoughts on that?
Yes, potentially, but I think that right now what we are thinking and what we know is that the enzymes that are involved in the CAM pathway are conserved in C3 plants. They just, for example, phosphoenol pyruvate carboxylase is involved, I believe, in sugar metabolism in C3 plants. In CAM plants, it just has this very important role in photosynthesis. So the idea is that as opposed to having novel genes that make the CAM pathways, genes are just repurposed in CAM. So we are thinking that if it is one transcription factors or two or just a bunch of them that actually help to activate enzymes at some point, because obviously C3 and CAM are compatible because the ice plant exists and is able to accommodate both within the same cell. So we're thinking that it will take for sure the introduction of several genes, but potentially not too many of them. I think the bigger question right now is that CAM plants, for the most part, are succulents and so have very thick leaves. And so it will be interesting to see if plants that have very thin leaves, like cereals, are going to be able to accommodate this pathway. But there's actually some studies showing that succulent is not a prerequisite for CAM. So I don't know. We just don't know enough about it for the moment.
Are there any other questions? You have one. Sorry.
Thank you. Thank you for the presentation. No, it was really great. I have a question, it's just a technical question, because I'm not familiar with the techniques. Why is that you have such a large difference in the number of cells of nuclei that you have between I think it was between dark and night, that you have like twice in the night compared to the day? Either because any technical issue, is because the extraction method, which is either an explanation for that, or it's just random variation?
It's generally accepted that the number of nuclei will not be very homogeneous between samples. Generally, what is considered right now a fair amount of nuclei for a sample is at least 2,000, but 3,000 is really the threshold, the low threshold for significance. So I think the sample for which we have the least nuclei is 4,000 nuclei. We always load 10,000 nuclei. I mean, we load as much sample needed as theoretically we would need to get 10,000 nuclei. But for some reason, I've never seen a study achieving really 10,000 nuclei with one sample. I think it's mostly due to technical factors. The technique is still very new, but we actually noticed that our dog samples have a bit less nuclei. And we're thinking that it could be because of the acidity in the cell of the ice plant that is much higher. The pH is much lower. The acidity is much higher at night. And it could interfere with our nuclei isolation protocol. We don't really know, but this is definitely something we've noticed. For the moment, I don't think it's a big concern.
Thank you.
Thank you very much, Noe. Please join me in welcome Noe Perron for this final talk.